do you want to start? Oh, there we go. Perfect. On the same page there. Let me see. So I'm going to go ahead and share my screen. Oh, actually, I need to do one thing really quickly first here. Okay, I'm going to share my screen. All right. Can everybody hear me? I just speak up if it's not clear for any reason.
I can hear you.
Okay, great. Great. Well, again, thanks so much for having me. So I'm Hannah Whitehead. I'm the Honeybee Extension Educator at UMass Amherst. And I'm going to talk to you about some research that we did last summer with some queen breeders in Western Massachusetts and Eastern New York State, looking at some different methods of queen production and looking at queen quality. And I think the results we found were, I found them unexpected and kind of interesting and very promising as we think about how to rear more queens in northern climates. And so yeah, I hope you'll find it interesting as well. Before I get into it, I'm just going to look at my watch to keep an eye on the time here. But before I get into it, I want to make two quick announcements. One is this summer, we're going to be participating in the USDA APHIS honeybee disease survey again. So this is something that that is done at a national scale every year. The honeybee lab in Maryland works with inspectors and extension agents in different states. And those inspectors and extension agents will send honeybee samples to the lab in Maryland, and then they get added to this big national survey that happens every year so that we can track pests and diseases among honeybees across the country. So it's a really important monitoring effort. And we're looking for beekeepers for this summer. So if you have over 10 colonies in one location, you qualify to participate. And it would basically mean that I would come out, do a hive inspection, and then gather some samples and send them off for Varroa nosema virus testing, and then potentially for pesticides in pollen. So it's a cool opportunity to get some data on your hive and to participate in a big national project. So if you're interested, you can find my email at the bottom here, hwhitehead@umass.edu. So I encourage you to email me there. So yeah, and then the second announcement is that we're doing a project. So a couple of years ago, we did a project where we worked with 40 different hobbyist beekeepers and gathered all sorts of information about their hives, including information about pesticides in wax and in pollen. And we found some really interesting results, and beekeepers were really interested in it. And they also had a lot of questions about pesticides in honey. And the good news is, from the literature, we don't expect to find many pesticides in honey because pesticides, we know, tend to be lipophilic. So they'll bioaccumulate in places like wax and not honey, which is water-based. But we wanted to follow up on those questions. And so we're doing a small research project this summer where we're going to be collecting honey from 40 different backyard beekeepers in Massachusetts and looking at pesticide levels and land use, and also comparing it to honey from outside the state. And so if you're interested in getting involved with that, I would recommend, I'm going to put a link in the chat here. And I've got a little bit of info on the bottom of this slide here for people who can't see the chat. And so I encourage you to go to our website and click on the link to this project. And there's a place there where you can click on an interest form if you want to get involved. And right now we actually already have 40 beekeepers signed up, but there's plenty of spots. I'm going to create a wait list. And so that way, if people aren't able to participate, I'll open it up to people on the wait list and you can get involved. So I encourage you to check that out. And now I'll get started on this talk. So I'm going to start with a quick roadmap to orient you and let you know where I'm going with this. So I'm going to start with the context and why we decided to do this research. Then I'm going to talk a little bit about the queen life cycle and queen rearing. So this is going to be a little bit of back to bee school for many of you. Some of you maybe already reared your own queens. For some of you, this is totally new. This will make sure everybody's on the same page. And this won't be a queen rearing how-to, but there will be a lot of information about queen rearing. So hopefully it can whet your appetite to learn some more. Then I'm going to talk about our methods with this study and what we did. Then I'll talk about our results and what we found. And finally, I'll talk about some implications and future directions for this research context. So as beekeepers, you all know that high quality queens are really important for healthy hives. And you also know, or we also know from research that northern queens are important for northern hives. So there was a study conducted in Maine a couple years ago where they found that northern adapted queens survived winters, or that hives headed by northern adapted queens survived winters better than hives headed by uh, southern queens brought from the south. And so we know that there's all these potential advantages to having northern genetics in colder climates like we have in Massachusetts. And there are other advantages to having local queens too. So we know that importing queens, especially when we import them as part of packages, we can be importing pathogens and parasites. We know that shipping can have effects on queens and their vitality and reproductive potential. So there's a lot of reasons why there are advantages to raising local queens and to buying local queens. And if more beekeepers bought and raised northern queens, it can improve the sustainability of beekeeping in our region in northern climates like Massachusetts. But in order to have more local queens, we have to rear more local queens. And when we think about queen rearing, I think it's really helpful to think about queen rearing on a spectrum from control, from techniques that give you a lot of control over rearing conditions, but they're really difficult to do, and techniques that are easier to do, but don't give you less control over rearing conditions. So on the far left end of the spectrum, we have kind of the traditional Doolittle grafting method. So this method was invented in the late 1800s, and it's the most common way for people to rear queens today. It's sort of considered the gold standard for queen rearing. Many of you may have tried this out before. And so the advantages of this method are that you really are controlling larval rearing conditions, and you're setting these really optimal conditions for queen development. And the other advantage is you can produce lots of queens. The downside is that it can be really labor and resource intensive, and it can be um, really intimidating for newer backyard beekeepers to do. And on the other end of the spectrum is another technique that probably most of you have tried, which are walkaway splits. And so walkaway splits, the advantage of them is that they're easier and they require less resources. The disadvantage is that you have less control over rearing conditions. So, right, so you're pretty much splitting a hive. Anyway, I'll get into it a little bit later. But yeah, you have a lot less control, and so it's a lot of beekeepers. We'll talk about how when you do a walkaway split, you're actually rearing subpar queens, and I'll also get into that a little bit later. But that's kind of the potential disadvantage of this method is you're not controlling for larval conditions in the same way. And so I was approached in the spring of 2019 by two beekeepers, Ange Roll, who runs Baykeep Bees in Montague, Massachusetts, and Sam Comfort, who runs Anarchy Apiaries in the Hudson Valley. And they both were really interested in this question of how do we get more northern beekeepers in Massachusetts? Or I'm sorry, northern bees, northern queens in Massachusetts. And they were interested in both how do we help scale up people who already rear queens in this area, and how do we help backyard beekeepers raise their own queens? And so they were really interested in this question of walkaway splits and whether it's true that walkaway splits really produce subpar queens. And they were also really interested in this other method of queen rearing that kind of, I think, falls in the middle of the spectrum from control to ease. And that is what I'm going to call 48-hour queens. I mean, it's basically an abbreviated grafting method. And it's possible that this method has kind of the best of both worlds. It controls larval rearing conditions like a traditional 10-day Doolittle grafting method, but it's easier to do and more accessible, more like a walkaway split. And so maybe we get sort of the advantages of both ease and control and precision. And so 48-hour queens, I'll get into more what that is in a minute, but it is becoming sort of popular in queen rearing circles. And so what Ange and Sam wanted to test was whether queens reared in these two easier methods, 48-hour queens and walkaway splits, do they produce queens that are of comparable quality to the queens produced through the gold standard Doolittle 10-day grafting method? So can we produce high quality queens using easier methods that are more accessible to more beekeepers? And so we wrote a grant together and got funding from from Northeast SARE to do this research in the summer of 2020. And so before I get into this research, I want to quickly go over a little bit about the queen life cycle and why sort of the different points at which um, different points that impact queen quality in different ways and why we might think these different queen rearing methods would influence queen quality in different ways. So first, as all of you know, the queen bees can lay two kinds of eggs. She can lay a diploid egg that has two sets of chromosomes that will become female, and a haploid egg with one set of chromosomes that becomes male because they have this haplodiploid system of sex differentiation. And we all know that the haploid male egg is going to grow into a drone, and the female diploid egg has two paths that it could go down. It could become a worker or it could become a queen. So let's zoom in on that female egg here. And so that female egg, she's going to be an egg for 3 days, and then she's going to hatch into a little larva. And that's when she's going to kind of have this decision point where she can be tracked in one of two different directions. So if she is fed a diet that's a mix of royal jelly, pollen, and nectar, she's destined to become a worker. And you can see those worker larvae here on top. And if she's fed a diet, a lavish diet exclusively of royal jelly, she's destined to become a queen. And so these worker and queen larvae that are fed different diets will continue to grow, and eventually they'll be capped as pupa. And you'll all recognize on the top here these worker pupa, which are horizontal in their cells, and the cappings are flush against the frame. And you'll recognize these queen pupa on the bottom that are the kind of peanut-shaped wax shapes that are coming out from the comb. And so after they pupate for a little while, both the workers and the queens will emerge. And so for the workers, this whole process takes 21 days. For the queen, it takes 16 days. And then the queen, once she emerges, she will go back and try and kill all of the other queens that have not yet emerged. She's not always successful. Sometimes you do get 2 queens or more in your hive, but she'll at least try and do that. So she's the only one left. And so then if we zoom in and look at what happens to the queen after she emerges. So she emerges as an adult, and then she spends a couple of days becoming reproductively mature. And then she goes on a mating flights for about 2 weeks. I mean, this is really important because this is the only time in her life when she's going to mate. And what she does is she stores that sperm in an organ called the spermatheca that's in her body. And so then after these 2 weeks, she's done mating for the rest of her life. And she's stored all the sperm that she's going to carry for the rest of her life. And so now you have your mated queen. And she's going to come back to the nest and begin laying eggs, which she'll do for the next 1 to 3 years, depending on how long she lives. And so all queens are going to go through the same life cycle, but not all queens are the same quality. As you know, and you're probably familiar with, some queens are higher quality than others. And so first I want to clarify what I mean by high quality. So what I mean by queen quality is queen reproductive potential, which is really the ability for a queen to lay lots of viable eggs for a long time. And I think what's really interesting about queen honeybees is that unlike female animals from other organisms where reproductive potential is limited by the amount of eggs that they carry and the quality of those eggs, for queen honeybees, it's kind of counterintuitively, when we talk about queen reproductive potential, we're actually talking about the amount of sperm that she stores and the quality of that sperm. And so when we talk about queen quality, we're really talking about sort of 2 main things. And one is actually queen size. So there's correlations between queen size, spermatheca size, and the quality and quantity of sperm that she can store. So kind of her body type and physiology. And then the sperm itself. So how much sperm is she storing? How viable is it? How diverse is it? Things like that. So again, when I talk about queen quality throughout this talk, I'm really talking about these 2 things: queen size and physiology, and then the sperm that is stored in her spermatheca. And so what influences queen quality in the queen's development? So the most important thing that influences queen quality is the age at which she is tracked to become a queen. So there's a lot of research showing that the younger a larva is when it starts to get that queen treatment, that queen diet, the more queen-like it will become. And when a larva is tracked to become a queen when it's older, say 3 days old instead of 1 day old, if it's fed that worker diet for a couple of days, it will be less queen-like. And so what do I mean by queen-like? So these queens are bigger. And then they have all sorts of other qualities of reproductive maturity. So larger spermatheca, greater ovary weight, greater size and number of ovarials, greater number of mating flights, greater mating success, greater amount of stored sperm, and greater viability of stored sperm. So there's a lot of advantages to being tracked as a queen when you're younger. And I'm going to summarize all of these different characteristics as more viable sperm. So these more queen-like queens, they're bigger, they have more viable sperm. They literally look more like queens, right? They're more distinguishable from workers. And why do we care about that? Well, we care about that because she's going to be able to lay more viable worker eggs for longer. There's less of a chance that she'll become a drone layer, that she'll just stop, that she'll have a spotty laying pattern, all these problems that we don't want as beekeepers, right? And then why do we care about that? Because that means we're going to have a healthier hive with more bees, more food, more resources, all of these things that we like to see. And there's a lot of research correlating the age at which the larva is tracked to become a queen, and queen size and queen reproductive characteristics, and then hive quality and these other characteristics of hive success, like food storage and worker bee population and things like that. And then the other thing I want to point out that affects queen quality is the rearing environment. So there's also research showing that there are advantages to be raised in a rearing environment that's really nutritionally dense. So if there's plenty of pollen and plenty of nurse bees, those nurse bees can turn that pollen into plenty of royal jelly to feed those queens a really good diet. And there's research showing that queens that are reared in more rich and abundant rearing environments are more attractive to workers and have a greater acceptance in a new hive. And so we know that queen rearing conditions are critical for producing high quality queens. But our question is whether can we produce high quality queens using easier methods? So what is the queen quality like with a walkaway queen compared to a 48-hour queen compared to a 10-day queen in terms of queen quality or queen reproductive potential? So now I'm going to get a little bit into these 3 different queen rearing techniques and why we might think they would affect queen quality in different ways. So let's start with a 10-day technique. So again, this is the traditional Doolittle grafting method that's used by most queen breeders. So I'm actually going to lay out kind of the basic way that this is done, but I want to point out that there's lots of variations, and I can kind of point them out as I go. But there's many ways to do this, but this is kind of the basic framework. And so what you would do if you were doing this kind of method is you would first pick the hive that you want to rear daughters from. And you would do this sometime in late spring or early summer. So after there's resources in the environment and the colony producing drones, but not too much after the summer solstice. And then you'll take a frame of young larvae. So ideally only about a day and a half old. So actually the larvae in this picture are too old, but I wanted to get a photo that you could see. And then you will take your grafting frame, which is a sort of an empty frame with some bars and little grafting plastic or beeswax grafting cups hanging down vertically. And you'll transfer with your grafting tool, you'll transfer the young larvae from the original cell into the grafting cup. And so it'll hang vertically down in the grafting cup. And then you'll place that grafting frame into your queenless starter box, which you've already prepared. And so this is a nuke or a small box with a bunch of nurse bees and pollen and nectar, but no queen. And after about 48 hours, you can take that frame out and you'll see that those nurse bees who are eager to have a queen have started to draw out those queen cells. And so they'll start putting wax around the outside and they'll start feeding them with royal jelly, like you can see in this drawing here to the right. And so at this point, what you'll do is put it in a nutrient-rich finisher colony. So this is a colony that, again, you've stocked with a lot of nurse bees, you've stocked with pollen, you've stocked with nectar. Um, this is one of those points where there's a lot of different ways that people will do this. You can have a queen right finisher, a queen less finisher. Some people will combine a starter finisher. Um, there's a lot of sort of different ways to do this. But what the basic goal of the finisher colony is to have a super nutrient-rich environment, packed with nurse bees, packed with pollen, where you're putting these young queens to be fed as they grow. And then after about 8 more days, you can take your frame out, and hopefully you will see a frame that looks like this with a bunch of these peanut-shaped queen pupae. And then you will take them and place one pupa each in a mating nuke. And so these are small hive boxes or nukes that you've made up that have plenty of resources and nurse bees, but no queen. And so you'll leave those pupa in the mating nukes, and the queens will emerge, but you won't check them. And then after about 2 more weeks, during which they'll hopefully be going on mating flights, you can open up those mating nukes and hope that you see eggs and a mated queen. So that's the whole process. And I want to point out a couple of things about this process. So first, you're really controlling for larval rearing conditions. So you're selecting these really young larvae here, these 1-day-old larvae or 1.5-day-old larvae, to make sure you've got optimum conditions for your queen to grow. You're creating a really nutrient-rich starter box and finisher colony to make sure to give those queens the best possible nutrient environment. So you're really controlling for those conditions to rear high quality queens. But I also want to point out that you may have noticed that this is a really complicated process, and it involves actually a lot of bee resources. So you have to have lots of frames of pollen, lots of frames of nectar, lots of queen bees in order to make this whole process work. And it's pretty technically complicated. So it's not very accessible for newer beekeepers. I also want to quickly point out sort of the 2 points in the process here where the queens can be moved. So when they're in the finisher colony, they're actually pretty vulnerable when they're young pupa, and it's really not good to move them at that stage because you can really injure the queen. But when they're older, sort of later stage pupa, they're safe to be moved again. And actually when they're in that 48-hour stage, when you transfer them to the finisher colony is the other window where they're more resilient and you can move them around. Even though you would think they'd be really vulnerable, they're actually kind of stuck up there in that royal jelly and they're pretty safe to move. So you're kind of taking advantage of these 2 sort of windows of resiliency for your young queen. So now I'll walk you through the 48-hour or abbreviated grafting technique. So you'll see that it's pretty similar to the previous one. We're just going to kind of cut it off a little bit sooner. So in this method, you would also choose a colony to rear a queen from. Again, in the same late spring, early summer time of year. You would grab a frame of young larvae. You would lift those young larvae up with your grafting tool and put them in your queen cups on your grafting frame. You would put that grafting frame in a queenless starter box. And then after about 48 hours, you would take it out of the starter box and see that the bees have just started to draw out the wax and feed the larva royal jelly. And now, unlike in the 10-day method, you're actually going to take those 48-hour-old queens and stick them directly in mating nukes. And then you're going to wait a little while, so it'll take about 10 days for those queens to emerge. And then they'll go on mating flights for about 2 weeks. And at that point, you can check your mating nukes and hope that you find eggs and a mated queen. And so you can see from this that this process also controls for larval rearing conditions, but a little bit less than the 10-day process. So you're still selecting really young larvae that are ideal age to become queens. And you're still creating a nutrient-rich queenless starter box. But now you're transferring the cells directly to mating nukes when they're a little bit younger. And so you don't have that nutrient-rich finisher colony. And so hopefully you're building up some strong mating nukes. But you probably won't be as attentive to, your 15 mating nukes as you would be to your single finisher colony. but it makes the process a lot simpler. It means that it's only a 2 or 3 day process from start to finish. So it's a lot easier for queen breeders to get a lot of queens coming through. It means you're going to have a longer brood break in your mating nukes or in the hive that you put that 48-hour cell into, which could be good for varroa mites. So there's all sorts of potential advantages to trying out these 48-hour queens. But what we don't know is whether they will be as high quality as the ones produced using the full grafting method, because you are kind of cutting off the method about halfway through and not giving them as rich of a nutrient environment. And then the third method that we were interested in looking at are walkaway splits. And so again, there's a lot of different ways that you can do this and a lot of different variables. So I'm going to give you kind of a brief overview of kind of the basics of how it's done. But again, you're going to choose a colony that you want to rear daughters from during this same time window in late spring and early summer. You're going to split the colony. And so you're going to take, you're going to split apart those two boxes if we're thinking about this colony on the left here. And you're going to make sure that in each box you have resources. So you're going to make sure you have honey or nectar, pollen, brood that's going to emerge soon, and nurse bees. Plenty of nurse bees. And then the queen is going to be in one of those boxes, and then the other box will not have a queen. And you're going to make sure that in the box without a queen that you put a frame of really young larvae that are the age, really young, sort of 1.5 day old larvae that you would want to become a queen. And it's also really important that the box without the queen, you're going to stock really well with nurse bees, especially if this is the box that's not in the original hive location. So that's one of the tricky things with these walkaway splits is that, if you're able to move one of those boxes over 2 miles away, that's great because then you're more likely to retain all the bees in that hive. If not, all the foragers are going to go back to the original location. So it's really important to really stock that hive with a lot of nurse bees to give it the best possible chance. And so then you're going to wait about a month. And at the end of that month, hopefully you'll check your colony and you'll see eggs and a new mated queen. And walkaway splits have other advantages. So they can help with swarm control so that the colony doesn't build up too much too fast. And they also provide a really long brood break. And so they can help cut that reproductive cycle for varroa mites. But one of the questions about walkaway splits and about inducing that emergency queen rearing response in general is this question of will they rear high quality queens? And again, there's this kind of conventional wisdom that walkaway splits produce subpar queens that aren't as good. And a lot of people wonder whether, because what happens when you take the queen out of the hive, the bees will try and build queens from many different larvae, right? So that they have maximum chance to have a new queen. And some of those larvae will be older than others. And the larvae that are older are closer to being finished with development. So they're going to emerge as a queen first. And the theory goes that if you pull out the queen and induce an emergency situation, you're going to get a subpar queen because you're going to get a queen reared from the oldest larva, or from the oldest larva that makes it to emergence is going to be the next queen. And so that's kind of the general idea. But there was actually some research done where they looked at this and they actually found that the queens went through and they tore down pupa that were reared from larvae that were too old. So it's possible that even though some of these larvae are too old to become good queens, that the bees actually go through and selectively tear them down, and they select for the queens that are reared from the correct age larva. And so, but there hasn't been that much research on this. This is just from one study. And so there's a lot of kind of this open question about can you rear high quality, essentially emergency queens? And I want to point out that in this research, we weren't actually making true splits. We were actually simply removing the queen from a mating nuke in place. So we would come in and simply remove the queen. So I just want to point out that that's kind of the other ones pretty closely mirror our techniques. And in this one, we didn't make a true split, but we simply removed the queen to induce that emergency situation and then left the mating nuke alone. So we can come back to our spectrum from control to ease for these three queen rearing techniques. And now we can really picture how they fit together along the spectrum, and come back to our original question, which was, can we produce high quality queens using easier methods? And how does queen quality compare between these three methods? So now I'll get to the methods here, and maybe since we're recording, maybe I will get all the way through and then take questions at the end, if that works for everybody. Feel free to interrupt if you have a burning question. So now I'll go through our methods and what we did last summer. What we did is we reared queens through these three different methods. And Angie and Sam both have robust queen rearing operations, so they were able to really scale this up and rear a bunch of queens in these different ways. So we reared in total about 100 queens in each method. And we did this in two different rounds. One in sort of late May, early June, one towards the end of June. And in each batch, all of the queens from that batch were from the same genetic line. So we were controlling for genetic differences. And then what we did with these three queens is we were really interested in comparing queen quality. And so this research, we didn't end up taking this all the way sort of to the end result. We didn't end up putting these queens in new hives and then looking at these new hives, since this was a preliminary one-year study. But we were really interested in looking at metrics of queen quality to know whether it even made sense to do a longer study. And so what we measured were in the field, we measured queen take, so percent take in the mating nukes for these different queens, or how many were there when we checked to see if they'd successfully mated and began laying. And then we measured the nuke population size. And then we took a subset, so 51 of the 314 queens we sent off to the Tarpey lab in North Carolina, where they were analyzed for a morphological grade, which combines things like head width, thorax width, and weight. They were analyzed for insemination grade, which includes things like spermathecus size, amount of sperm, amount of viable sperm, things like that. And they were also assessed for an overall grade that included all these different metrics. And so what we predicted based on the literature and based on sort of conventional beekeeping wisdom is that queens reared through the traditional grafting technique would be of higher quality than queens reared through the 48-hour technique, and higher quality than queens reared through the walkaway technique. And so some quick photos from our research. So this is Sam Comfort's, one of Sam Comfort's apiaries out in the Hudson Valley. And each one of these stacks is actually several different nucleus colonies. Can everybody hear me? I got a message saying my internet connection was unstable. So we'll holler if I start breaking up.
Will do.
Okay, thank you. So I've got, so you can see he kind of stacks these different, each box is its own mating nuke in these little boxes. So this is out again in the Hudson Valley, and you'll see a close up here. So here's Ange on the right and our research assistant on the left, and they're looking through the colonies, looking for queens and looking at population size. And here's Sam looking for a queen in one of the colonies. As you can see a little bit more into these cool little warrior hives that we use for mating nukes. There's a little bit more of a close-up of Sam grabbing a queen and the hive there. And then here's some of our queens ready to be shipped off to North Carolina. Great, and then that's, so now I'll go a little bit into our methods and what we found. So I'm gonna start with our field metrics here. So in terms of queen take, and again, what I mean by queen take is basically queen survival. Did she make it to be an adult mated queen? Did the bees accept her? And so on the right, we have our graph, and you can see on the x-axis, the three different treatments. So the 10-day, 48-hour, and walkaway treatments. And on the y-axis, we have queen take, so percent queen take, what percent of the nukes do we have a queen success? And what we found was we found no significant difference between our different groups. They were all sort of 65 to 68% successful in with their queen take. So there was no statistical difference. And then if we look at nuke population size, again, we have our 3 treatments, 10-day, 48-hour, and walk away along the bottom. And on the y-axis, we have average nuke population size rating. And so that's from 0 to 3. So we rated it a 3 if the nuke was bubbling over with bees. It got a 1 if it kind of had a sparse population, and a 2 was somewhere in the middle. And we found that the population for the walkaway splits was significantly lower than the 10-day and the 48-hour cells, which was not surprising, given that we know that the walkaways have the longest brood break. And we know that that's something to watch out for when you're doing a walkaway split, is you are creating a really long brood break that's going to impact the population. But it was interesting to see that in the data. And so if we look at our field results overall, we found that queen survival was the same across all 3 groups, and nuke population size was a little bit smaller in our walkaway split queens. And then if we look at our lab metrics, so we can start with morphological grade. So again, we've got our graph on the right with our treatments along the bottom. And then we've got morphological grade on the y-axis from low to high. If you look at the box on the left, you can see, just as a reminder, what that includes, which are things like waist, head width, and thorax width. And what we found was we actually found no significant difference between our 3 treatments in terms of morphological grade. And then if we look at insemination grade, so again, we have our 3 treatments along the bottom of this graph, and insemination grade from low to high on the y-axis. And you can see in the box on the left, that includes things like spermatheca size, viable sperm count, and total sperm count. And again, we found no significant difference between our 3 different groups. And so then if we go down to overall grade, which incorporated metrics from all of these different, both insemination and morphological metrics. Again, the treatments are along the x-axis on the bottom, and overall grade from low to high on the y-axis. And again, we found no significant difference, which again was not surprising at this point, since we found no difference with the other 2, but in general was a really surprising result. I'll let you look at that for a moment. So then overall, what did we find with our lab results? So if we're comparing our 3 different treatments, we found that morphological grade was equivalent, insemination grade was equivalent, and overall grade was equivalent. And so what does this mean? So I think there's a couple of main takeaways. The first was that all the methods produced really similar queens. And I think normally in science, when you get a result that everything is similar, it's the result you least want to see, because it means that you haven't found anything interesting. But this is a kind of unusual case for finding that all 3 methods were really similar. It was actually really surprising. We were all very surprised. We thought at the very least the walkaway queens were going to be inferior to the other 2 grafted queens. And so I think this really made all of us wonder whether we should be paying more attention to walkaway splits and doing more research on walkaway splits and exploring more about their potential, since they're the easiest way to produce queens and are especially suited to backyard beekeepers and newer beekeepers. And then the second takeaway that was less surprising was that when we did the walkaway splits, or when we removed the queen, the worker population was reduced relative to other methods. And so this, again, was not surprising. We know that they experience the longest brood breaks. We know that that happens when you create a walkaway split. But it's just a good reminder to be aware that, even though that method is easier and they produce equivalent queens, to keep in mind that the population really takes a hit. And so to make sure that your hive can handle a really long brood break. And so future directions. So I think one interesting question about this research has to do with how environmental conditions affect these results. So it's possible that this was a particularly good spot in the Hudson Valley where we were doing this research, and particularly good conditions in May and in June. And it's possible that if the conditions were worse, you would start to see more advantages from grafting techniques. But we don't know. So that's an open question that could be explored. Another question that I think is a really important question to explore, excuse me, is what is the best way to make a walkaway split? So there's a lot of different factors, as many of you have probably explored as beekeepers, right? So again, do you move the walkaway split 2 miles away? How many nurse bees are you adding? Do you leave the box with the original queen in the original location, or do you leave the box that doesn't get a queen in the original location? How are you stocking those hives with resources? All sorts of, when in the year are you doing this? All sorts of questions like that. And so I think those questions are really important to answer, because sometimes what influences whether or not a walkaway split is successful is not just whether or not it's possible to raise a good emergency queen, but is how are those splits made, and making sure that they're given the most resources to have the biggest chance of having a successful queen. And I think there's a lot of research to be done around that. And then finally, the most important thing is to look at the effect on laying pattern and colony health, right? Because at the end of the day, as beekeepers, you don't really care if your queen is big as long as she has a healthy colony, right? And so we know that these metrics of queen quality are often correlated with hive health and with hive success, but we don't know that that's the case in this instance. And so it would be really important, or it seems really intriguing to plan a longer research project where we could, again, compare these 3 types of queen rearing, but follow them all the way through till we put the queens in full-size hives and look at laying pattern and colony health, and see if they're equivalent between these 3 methods. So that's another big question mark with this research. But overall, yeah, we were very surprised by these results, and it led to lots of questions about whether we should be exploring more research on these other methods, and figuring out ways to make them more accessible to beekeepers and more foolproof for raising high quality queens. And so I want to acknowledge Anarchy Apiaries and the Keep Bees and Northeast SARE for helping out with this research. And here's my contact information, and I'm happy to take any questions.
Hannah, is one of your future questions also to look at the longevity of some of the queens? Because I've found over time with my walkaway splits that time of year and so on, that there's really something to, will this queen produce into her 3rd year? I like to keep them for their 2nd year. They're the most productive, and then the next summer I split that hive and I replace that queen and so on. Could that be one of your questions too, or are there enough studies on that?
There absolutely could be. I don't know of any. I mean, I haven't read any studies about queen rearing conditions and queen longevity, but that's a really good question, especially because I know that more and more beekeepers are having issues with queens not being viable for as long, and we don't quite know why. And that's obviously a really important metric of, what we want to see in a good queen. So that's a really good, yeah, we haven't, yeah, that could definitely be incorporated if we were doing a larger study. It would be really interesting to look at. Yeah, that's a good question.
Yeah, very good presentation. Thank you.
Have you found that your walkaway queens don't live for as long? Or have you just been wondering?
No, it really depends on the base stock that I have. At the end of this year, I'll have more kind of definitive answers to that.
Yeah.
But uh, I have one line that I've been running since my first queen, and sometimes they're really good, and sometimes I don't know why they fail. So
That's
it's
interesting.
and it's a strong line. It's not very productive, but they're really nice bees, so I like working with them. So it's in that way.
That's always good. Yeah,
So yeah,
that's great.
thank you.
Yeah.
Yeah, I have
Yeah.
one or two questions.
Yeah.
One is that I expected you to say, and so we've come to the conclusion that the 48-hour method is the best. I mean, or something like that, because you were sort of
Mhm.
leaning that way in the beginning, and then toward the
Mhm.
end, you didn't follow that up. So any comments on that?
Yeah, that's a great question. And that's kind of that kind of followed our thinking with this. I mean, we originally just sort of this what's the word, designed this as a research project to test 48-hour queens. That was kind of the whole framework of this. And we were testing walkaways as kind of a almost like a negative control. We were really interested in 48-hour queens.
Right.
And the fact that we found that the walkaways had this, very similar metrics to the other queens led us to wonder, well, maybe we shouldn't even be looking at this intermediate method. Maybe we should be thinking about these walkaways. But I think that was just the most surprising thing for us. And so that was what so one of the most interesting takeaways for me. But I think you make a good point that, and I actually think that the 48-hour method and the walkaways have a really different role, because walkaway splits are really much more useful for backyard beekeepers, but they're
Mhm.
not as useful in helping small scale queen rearers, right? But if the 48-hour queens are also really effective, that could be a way for queen breeders to rear queens more quickly, get those cells out to beekeepers. Those beekeepers then have a longer brood break when they put that 48-hour cell in, and they kind of learn more about queen biology. I know queen breeders are really excited about 48-hour queens as the potential to kind of exchange genetics more easily and more rapidly. Um, so yeah, I'm glad you brought that up, and we can the fact that yeah, this is also really promising for thinking about 48-hour queens as a potential technique.
And one other
That answers your
question, this is
question.
kind of a
Yeah.
beginner question. Say we're talking the 10-day grafting thing, and you get graft into a grafting frame, and then you take those cells and put them, well, somewhere. But my question is, what is it that triggers the bees to treat this larvae as a possible queen as opposed to a worker bee? What triggers their action that way?
That's a great question. And I don't know exactly. I've always assumed it's because they're placed vertically in the cells. And often when people do
Mhm.
grafting, I mean, there's different grafting techniques, but one of the grafting techniques involves placing it in a little bit of royal jelly in the bottom of that queen cup. But my impression is because they're placed vertically and you're putting them in a queenless colony that's sort of bursting with nurse bees, that they're really rearing and ready to raise a queen. You're not going to want, and also that starter box would also not have any other young larvae. So I think, the fact that they're vertical is going to be spurring them, but also the fact that there's no other young larvae. And so those bees are waiting to have a queen, and then you're putting in some young larvae and they're ready to go. If that answers that question.
Yeah, sure.
Yes, you wouldn't want to have a frame of 1-day old larva in that starter box, because then they might
Right.
not draw out your cells as much. Yeah,
Okay, thank you.
good question. Yeah. I'll quickly check the chat too. I haven't. Great.
Hannah, it's Bernie again.
Yeah.
I'm actually looking for a small scale queen production, kind of in between. I don't need 15 queens. I maybe need 4 or 5 a month or every couple weeks
Yeah.
to help friends out and start. So if you ever run across something that's a little smaller scale, I mean, I can offset making nukes or breeding nukes with, double nukes and taking out the extra frames of brood. And
Yeah.
that's my main experiment this year, to get
Oh,
more
cool.
comfortable with that. But I'm looking for something that's in between with less manipulation.
Yeah, that's a great question.
Yeah.
The only thing that came to mind, I don't know if you've heard of the close board technique.
Yeah. The problem is I don't know, the 10 frames seems like a lot. So I've been exploring some other
Mhm.
things that I'm going to try this year. There's a
Yeah.
smaller technique called the Morris board.
Oh, cool.
Beecraft puts out a little pamphlet on that, and there's
Yeah.
very little out there on it.
Yeah.
And
Oh,
like the
interesting.
videos on it are, yeah, it works, but they don't really show
Uh-huh.
you. And the person who has them he never does statistics. He just kind of tells you. I
Yeah.
don't trust
Yeah.
that.
Yeah. That's interesting. Huh. That's interesting. I
Yeah.
mean, I wonder if even the 48-hour method could kind of, if you're just kind of creating a starter box and practicing grafting and then putting
Yeah.
those cells directly in, new little nukes or splits or something. And I'm
Oh,
happy to connect you. Oh, go ahead.
once you have classes again, I'm definitely I'll be at your next grafting class, but
Yeah,
you know, once we're through all this. So
absolutely. And I'm happy if you are interested in learning more about the 48-hour technique, I'm happy to connect you to Andrew Sand, who really did a lot of um, the actual grafting
okay.
and clean room for this project in there. Yeah, they have a lot of knowledge, so they would definitely
Okay.
be happy to share. Yeah,
Well, thank you.
yeah,
Hi,
Thank you
Hannah.
very much.
yeah.
Hannah.
Hi.
I'm Juanita, and I just want to say how much I enjoyed your presentation. And
Oh,
especially
great.
the visual aids were really good. I'm a retired teacher, so I know how important those are. But I have 3 hives that were splits from a friend mentor of mine. And
Oh, cool.
they were originally they were dead outs from last winter, and then he split his, and they've boomed. They've done beautifully. I mean, they're all out flying now. But I'm wondering if I might actually split them. I've never done it before, but do they have to be, as I heard you say, 2 miles apart so they don't return to the old hive when you split it?
So, yeah, so ideally, if that's a possibility, that way, any of the foraging bees that are in your hive, they won't go back to the original location, and that way, you can have a high population in both of your splits. But that's often, in fact, probably most of the time, is not the case for beekeepers. You don't have another yard 2 miles away. And so the thing that's really important that I think is the pitfall that a lot of people, the trickiest part about making splits is just making sure to get a lot of nurse bees in the colony that's not in the original location, because those nurse bees that aren't flying yet will stay in that colony and hopefully um, keep it operating smoothly and keep everything going well until they get older and become foragers who only know that new location. So it's just really important to probably shake 2, 3, 4 frames of open frames of bees that are open brood that are covered in nurse bees into the box that's not in the original location.
Okay.
Yeah.
But it doesn't have to be that far away. I could do it in my own
No.
yard.
Yeah, if you could put it kind of on the other side of your yard. Yeah, I mean, all the foragers will go back to the original location, but if you shake enough nurse bees and it's early enough in the year, you should be okay.
Okay. Thank you. It's great.
Yeah, no
Enjoyed it.
problem.
I'd like to learn more
Yay.
about it.
Yay, that's great.
Thanks.
Okay, if no one else has any questions for Hannah, we will let her go. Thank you very much, Hannah, for a wonderful presentation. I got just as much out of it a second time around as I did the first, so thank you very much. And thank you everyone for attending. We will be having a members meeting this Friday night. We'll talk about the status of the club and looking forward to activities this summer and continuation of our speaker series. So we'll want your feedback on that. And then next Monday, we are lucky to have a member of the National Oceanographic and Atmospheric Administration to talk about weather forecasting. So we're looking forward to that as well. So thank you everyone for coming and have a good evening. Take care.
Thanks for having me, and thanks for all your good questions. It was great. It's kind of a fun to have a small group, actually.
Thank you, Hannah.
Take care, everybody.
Excellent. Thank you.